All right, we are on our next section, which is our visual system, which we're going to be looking at how do our eyes process the world around us. And it's one of our senses that we use the most. So it's quite interesting how it all works. All right, so we're going to dive right in to the visual stimulus and the eye. So light. So how do we see light? So light technically is a form of electromagnetic energy that can be described in terms of wavelengths, okay? Now, these light waves, right, or these wavelengths, they travel through space in our environment, right? So technically, if our eyes didn't work the way they do, we would literally see these wavelengths in our environment, okay? But we don't. All right. So these wavelengths basically, and I just want to show you this, guys, is the different waves that and rays that are in our environment right now. Now, these go from all the way from gamma to the AC circuits, and the only waves we could technically see are right there, right? So that is what we are able to process, which we'll look at that. Those wavelengths, it's what allow us to see color, okay? Or our brain processes those wavelengths and make them into color. Now, a wavelength, and you are going to need to know these waves. You're going to need to write these down and have them in your notebooks, please, because these wavelengths of light is basically the distance from the peak, right, of one wave to the peak of another, all right? Now, the way that those wavelengths of light happen or reflected is what determines the hue or the color. Now, there are other waves out there like radio waves, infrared radiation waves that we cannot see. Now, the amplitude is basically determines the brightness of the stimulus, okay? So if you look here, if it is a short wavelength, it's going to have more bluish colors. And these are going to be the same wavelengths you're going to need for auditory, okay? If it's a long wavelength, it's going to have more reddish color, okay? The amplitude, which is the brightness, right? The higher it is, the brighter the color. The smaller the amplitude, it's going to be a dull color, right? So if I'm wearing, I'm wearing a gray jacket, even though you can't see it because there's not a lot of light, so it's not able to pick up those wavelengths. All right. But this sure is releasing wavelengths that if the light, depending on where the light hits it, it would be able to see certain wavelengths. All right. Now, we're going to go into the structure of the eye, and I'm going to want you, if possible, to draw this. If you could print it, it would be great. And you're going to, along the lines here, kind of write what the function of all of these things are. Okay? So we're going to start with the first thing, and it's not actually on here, but it is what's called the... Oh, wait a minute. Matt, I should probably pause, but I'm not going to do that because I'm not. Okay, I think I'm going to say this right. Sclera, that word, it's ERA. And this is going to be the white outer part of the eye, okay? That helps us basically maintain the shape of our eye, and it also protects us from any injury, okay? Then we're gonna move on to the iris. And the iris is basically the colored part of our eye, and it contains a muscle that's what controls the size of the pupil, okay? So the iris, the colored part, is actually a muscle, and it's going to dilate and constrict in response to the light around us. All right, so it's not the pupil that moves in and out, it's actually the iris that's a muscle. Now, the pupil, it appears black in our eye, but in actuality, it's just the opening in the center of the iris, and it's literally an opening. That's why it looks black, because it's really kind of like a hole in our eye. And then it gets adjusted by the iris adjusts to the light in order for to determine how much light goes into the pupil. Okay, now you also have what's called the cornea. Now, these are the cornea and the lens are their job is to help us focus. All right. So the cornea is a clear membrane that is in front of the eye. All right. This is the one that does most of the bending with light. Okay. Now the lens, all right, which is here, is going to be it's transparent and somewhat flexible. And this is the one that's going to fine tune images. Okay. Now, this is where it's going to focus on incoming light rays into the image to the retina, which the retina is in the back of the eye. Okay. So the lens kind of like sends it back here, and then that's, you'll see where they're going to convert this wavelengths to a neural energy. Okay, then you have, so the retina. The retina is very, very important. It's a light sensitive surface that records the electromagnetic energy and then converts it to the neural impulses. So then it processes it and sends it to the brain. Okay, this is the most complex area in your eye. And we have about, so in the retina, you're going to see that it's going to have, excuse me, it's going to have these cells in there, and I'll give you a closer look on that. But I just want to kind of finish here. But I'll show you a close-up picture of what's happening inside the retina. Now, we're going to have inside there's going to be rods and cones, okay? You may want to write that down. And then the fovea, which is inside the retina, is going to be, it's basically the center, and it only contains cones, and it helps with visual task, okay? Now, you have the optic nerve here, okay? And the optic nerve is what carries the neural impulses from the eye to the brain, okay? And then from there, it sends it to the thalamus, and then the thalamus sends it, right? Thalamus is the relay station, sends it to the part of the brain, which is usually in the occipital lobe, to process what we just saw, okay? Now, there is a blind spot, and you definitely need to know that, and sometimes you hear it when you're driving, that there's the blind spot. And basically what that is it's where the optic nerve is leaving the eye. And in this little part right here, where it happens, where they meet, is where you will not be able to see, okay? So there's no receptor cells there. So write that down. There's no receptor cells. That's why you won't be able to see anything if it's in your blind spot. And there's a little test that you could see how far your blind spot is, or where exactly it is. Okay, so what I want to do now is I'm going to show you these visual receptor cells that are inside the retina. Okay, so if you can draw this out, kind of get a little, idea of what this looks like, of what's happening in the retina, that would be great. If you could print it, even better. But please note that this is visual receptor cells are in the retina, okay? And there are some cells that you need to know, okay? And the first one you need to know in here, the cells that are here, is the rods. So these rods, as you see here, okay, the rods are to detect black, white, and gray, okay? And they're sensitive to light, but they are used for the like shadows, and for us to see at night, okay? They're very good at night. This is the ones we have the most of. We have about 120 million rods, and this is in order for us to see clearly at night, a little bit better, right? And so um, they're gonna also share bipolar cells, and you'll see here bipolar cells with other rods. And that's what these are basically gonna be, the ones that send, they do the transduction and send it to the optic nerve to then the brain, okay? Now, the cones are receptor cells that are used to perceive color, okay? They work best in daylight and under high illumination or light. We have about 6 million cones, and they're going to all cluster around the fovea, all right? And then each of them is going to transmit to a single bipolar cell that then helps them relay that information to the visual cortex, okay, which is the converting it into a neural impulse, all right? Then we're gonna go into, okay, so in the rods and the cones, that is where there's going to be transduction happening. And these two cells, the bipolar cells, are going to work with the rods and cones, and then it's going to send to the ganglion cells. And then the ganglion cells, the one that sends that transduction, that neural electrical energy through the optic nerve to then the brain, to the back of the brain. And we're going to talk about visual processing and how it works, okay? So the way that this works is that once the signal reaches the brain, right, what's going to end up happening is those neural impulses, if what I see in my right side is going to go to the left side of my visual cortex, need to know that. And then you're also, whatever you see with your right eye, it's going to go to the left, I'm sorry, what you see with your, what you see on the left side goes to your right eye. And then that is what's going, it crosses over, sorry. I'm a little confused here, hold on. Okay, my apologies, it's a little late. All right, so remember we've talked about this. What we see on the left side, we saw this with the split brains. What we see on the left goes to our right, and that's on the side of, goes to the right side of our brain. What we see on the left goes to, right, it goes, what we see on the right goes to our left and the left side of the brain. And this is in the part of the visual cortex, okay, in the back, or also occipital lobe. All right, now there is also what's called feature detectors, and we've talked about this when we talked about the brain. And feature detectors are basically specialized neurons in the occipital lobe, okay, so this is all perception part, in which they're going to receive the information, and then they're going to look at specific features such as edges, lines, angles, and movement, okay? And a good way to like kind of remember this is SAM, which is shapes, angles, movement, and feature detectors are what basically processes this information, and that is located in the occipital lobe, okay? Now, we also have what's called parallel processing, and parallel processing is when we're able to analyze a visual scene, and what our brain does is basically divides it into sections from like color, motion, form, and depth. And it's doing this all at the same time. That's why it's parallel processing. And then basically it puts it together so that we see one thing. Okay, we're not seeing it in pieces. It puts it together, our brain puts it together, and then boom, we see a bird with all of these things, the color, the motion, the form, and the depth. And obviously this happens very, very quickly. Okay, and then there is also another concept here, and that is called binding. And binding is basically when our brains bring together and integrate what is processed, okay, by using different cells. All right, so binding is just basically putting it together. and making it make sense to us, okay? And let's talk about some problems in vision. All right, so vision problems, there's a few. And the first one is gonna be myopia, which is also called nearsightedness, okay? And this is when we have our vision is very good from near, but we're not able to see things at a distance, okay? So like I have myopia, I'm able to see really close, but not not far away. Then we have it's either hyperopia, it could be in two ways, or presbyopia. And this is basically farsightedness, that they can see good from far, but they cannot see items close by. All right. The best way to fix these two problems is going to be with glasses or contacts. They typically can correct that. In order to find out if you have any of these two things is to take what's called the Snellen test, and I'm sure you've all taken that because they do that in school, and you go to the doctor and you see a bunch of letters, they all have different sizes and from a distance, and then it's how far can you read on the line as it gets smaller. There is also what's called astigmatism, and this is basically where your cornea is irregularly shaped, okay, which causes blurriness at any distance. So it could be from close or far, they see blurry because their cornea is irregularly shaped. All right, then there's also what's called cataracts, and a lot of times this happens with people that are getting older, which is where the protein in the lens is going to start breaking down, and what it starts creating like cloudiness in the eye, and so the person starts seeing kind of cloudy their vision, and you could even sometimes see it in their eyes. And there is surgery for this what they do is they'll replace the lens, and then that restores the vision. All right, then there's conjunctivitis, which is basically pink eye, and this is where there is an inflammation in the conjunctiva, which is the transparent layer that basically lines our eyelid, and then it covers the sclera, the white part of the eye. Okay, that's pink eye. It irritates it. Then you have the last one, which is glaucoma, and this is basically damage to the optic nerve and will destroy the vision. There's no way of curing that, okay? Because the neural signals are not being sent to the what's this called? To the brain. All right. Now, there are theories on how we see color, okay? And you need to know these two theories. And since there's two theories, there's two ways of determining why these theories actually exist, okay? And so the first one is gonna be Young Hellman's, whatever that guy, trichromatic theory, all right? And this theory says that in the retina, there are only three cone receptors, and they are red, green, and blue, okay? And what they do, how this theory works, is that the combinations of these cones, they work together in order for us to see colors. So those wavelengths that are out there, they combine, and using just these three cones, it mixes together, they work together in order for us to see a variety of colors, okay? Now, in order for us to determine that this theory is accurate, okay, or it supports this theory, is people that are colorblind. So colorblind is basically that, and there's different types, and I'm going to show you here, there's different types of color blindness. It's not that they're actually like colorblind, that they don't see any color. It's just that some of their cones are not functioning properly. So it could be like the red, the blue, or the green, and then that will determine how they see shades of those colors. Like, so for instance, typically in color blindness, it's typically males and people with light eyes. So like my father, he's colorblind, and he can't see like good shade, like shades of, I think it's red or blue. And then so I ask him, and he says that he sees, like he knows it's a distinct shade like of blue, but it's not like the blue we see. So it's kind of like, he knows it's a different color, but it's not quite. It's like kind of different shade than we would see it. So there's different types, right? So here you see this flag. You could see somebody that this would typically, if you have no color blindness, you see the red and blue of the flag. This would be somebody that is missing like the red cones, or the red cones are not working as properly, so they won't see the red, but they'll be able to see like green in there, even though there's no green. And then you have, the variety there. You guys could see that there. Okay, so color blindness is what kind of supports this theory. Then there is the next theory, which is called opponent process theory. And we've talked about opponent process theory when it comes to emotion. This is one for vision. So opponent process is basically where we have receptor cells. The cones have, they work in pairs. Okay, so these cones are going to work in pairs, and they're red, green, blue, yellow, and white and black. Okay, they're going to work together. And the way that this theory works is that they will, one cone will they work in pairs, right? And if one is being used the most, right, the other one kind of will take a back seat until you use it too much, right? If I'm staring at something that's like red, or like in this, hold on a second. Sorry about that. My Siri's like all of a sudden hearing this whole thing. Like if I'm staring at this green, right? If I stare at it for a long time, that cone is going to get fatigued, and that's how this theory works. If one cone gets fatigued, then the other will replace it. So how do we know this to be true? Is after images. After images, it's called after images, is where we stare at something for a long period of time. It has a certain color, and then if we look away from it, then the other cone takes its place. So if you did this correctly, and you're not going to be able to do it so good here, because you would have to stare at this for a long period of time and then stare at something white, right? And it's kind of hard because this has the lines on here, but there's plenty online that you could just look for after images, little test. But you would stare at this little center and then stare at it for a long period of times. Those receptors of the green and yellow and black would get tired, and so when you look away, then red and blue would take its place. Okay, pretty cool. And it does work. So after images supports opponent processes theory, and we are done.